end up just classifying also the other points. Because we can develop some ideas to enhance the presentation of this class, but we end up misclassifying the points from the other class. One of the state art of the solutions to tackle this problem is called SMART, synthetic minority oversampling technique. Very intuitive and very simple idea. It's based on the following. So let's think about these red stars as the positive points, and these black circles are the negative ones. Now this idea is saying the following. Take Dr. Yusuf. Dr. Yusuf is the closest one to me. But see, there is a gap. There is one chair here. Okay? So we need one guy to come and sit here, but that guy who shares something common between me and him. So I'll sit down with him, agree on something, and develop some certain maybe characteristics or skills, and we ask one guy who has those skills to come and sit between us, because we need to maintain our group for the own goal, right? So this is the same thing. So we take one of the points, we take the closest point, and then we measure the distance, and we do what's called interpolation. We just generate a new sample based on the relationship of these two samples. And we do this randomly. We end up adding more points. So you can say I have only 10 points. Now you can use this method to generate 100 points, 20 points, 30 points, whatever you want. Okay? So I came here and said, there are limitations with this method, because I'm doing it randomly. And I'm claiming that these points are the points that we should give more support for. Why do you think so? You see, those points are very close to the negative class. There is a high chance to really misclassify those. So those are really my friends that I want to give them support to survive. But those who are already somewhere here, they are far from the negative ones. They're already in a safe zone. So I developed this idea to categorize this space into safe zone for safe cases and dangerous cases. And based on this quantification, now I'm able to control the synthetic generation. It's not anymore random, just we choose the nearest neighbor. Now maybe I have something more common with Ahmed, so I will choose him, and then I will come with a better fit for me in this space. So this is the idea we developed here. And then we took that method for this type of data that I'm talking about, that would help eventually in drug discovery, and we applied it. We compared it with the SMART, the same technique that we are using, and in the remote, a version that's developed in 2014 in one of the top journals, IEEE Transactions in Data Knowledge Engineering, that was shown to be better than SMART. And we were comparing with all of these types of state of the art. We were carrying actually, this is just a snapshot of the results of the paper. You find more data sets. We're caring about improving precision in our case. So here we improve precision. Maybe we have sacrificed some sensitivity, but we care more about precision. Why do we care more about precision? Because the moment I say for the bioscientist or the biochemist, take this molecule and test it, and he trusts me, and then he take it and then discover that this was false, it means directly that more money was spent and more time was wasted. So we don't want to end up doing this, and precision reflects this, actually. To take a summary measure so that we know that at least we are not sacrificing so much on that side, we see that in all cases we improve than the other competitors. Now we might think, oh, we came through some challenges. We were able to reduce the number of variables. We were able to handle this problem of class imbalance and solve the bias. And that's enough. Let's develop our model. When we deal with bioscientists, biochemists, and those experts in medicine, this is not enough for them. Because remember I told you in the beginning, even themselves, when they take some experiments to further clinical trials and phases, it might fail in the future. So they know that you might be saying that I have a good thing, then it might fail actually. So we wanted to do some more validation, borrowing some ideas from their side. So we did the following. We brought some drugs, some actual drugs that you can find in the market from the databases, And we took these drugs and we give it as input to our system. And then our system, towards the thyroid stimulating hormone receptor, the example I gave earlier, was saying, okay, there's one of the drugs that can be having some effect on this thyroid. So we took then this drug and we did one of the experiments that's known for the field of biochemistry as 3D doping, which means I bring the 3D shape of the protein. And this is an actual 3D shape of the protein. Except that it's static, but in our human body it's not static, it will be moving, right? And then we run some software. They are not our softwares. We give it the protein, we give it the compound, the drug molecule, the 3D structure of the drug. And now the problem is about doing some key and lock insertion. So we have the lock, which is the protein. And the time we fit the key in that protein, we can have an interaction. So in our human body, an interaction means some kind of binding, what they call binding in biochemistry. So these two things should bind. Otherwise, an interaction wouldn't be happening. And if you look, when we did this experiment, you see that there's a nice fitting here. And our drug was just directly fitting there, which gave a nice way of validating maybe over the results that we have. I want to show you now in this short video the effect of one of the rare diseases. It's called Niemann-Pick type C disease. It's a neurodegenerative disease which has no treatment so far, till now. So let's watch the video. So see this young girl, she's at the age of 6, and she had that disease. Now see at the age of 9, she's having some disabilities. And what I'm telling you, they're facing challenges with these diseases. So if one of the kids was diagnosed with that disease, we don't have a treatment. So these are some kind of the interesting cases where we can bring our solutions and see what we can get. There is another solution that's here. Given the limit of the time, I will not be describing the solution, but I can reference you to the published paper there. That solution will be actually playing on the optimization side based on correlating different data sets. And anyway, to make the story simple, we brought the drugs from the drug bank database, and we did again the same experiment that we did before. But now we're dealing with a different data set. It's the data sets, or two data sets. We're dealing with two data sets that relates to this Niemann-Pick type disease. So we found out that there are two data sets that they are talking about the same disease, but from different types of perspectives. They are dealing with different genetic information. And then we brought the drugs and we gave it to this model and system. What we have found out that one of the drugs were top hit for both of the data sets. So we were happy about this, because this drug was not only reported to be active on only one data set, but also on the other one, and both of them are talking about the same disease. So we got really excited. So we went back checking, and this drug is called thiamidazole, and it's used to treat the human body, maybe the human stomach, for some types of nematodes or worms that can grow inside, or maybe in animals. But we have found out, based on collaboration with some biochemists in our center, that this can be having actually an effect on this Niemann-Pick type disease. We went further and we checked in some other databases, and we found some relevant concepts. But this Niemann-Pick type C disease, if you remember the girl that we showed, the disability that she developed at the age of 9, was a matter of affecting some lipids or cholesterol movement in the human body. So the cholesterol does not move through the blood as we have it normally. And when we were checking some literature, we found that this drug has an effect on the cholesterol. It is the effect that goes in line with the story that we are proposing here. So in conclusion, in this type of problem for drug discovery, we went to one of the big resources, Optim database. It was released in 2009, and there was many research on that, but we worked heavily we produced some numbers that we can deal with in machine learning. We developed some ideas, as I showed you. Drama is one of them, to overcome limitations of other solutions that is tailored toward this problem. Because it's not only about improving the space in this region, but we want to improve the space, keeping in mind that we don't want to end up with a false positive that would cost more time and waste time and the money. At the end, we developed one more solution to bring up more data sets. That's how we were actually able to find a drug that was a common target between two different data sets with the same disease. So actually this work resulted in 3 journal publications, and 1 is ongoing under review right now. So 2 were already published, plus one in Journal of Bioinformatics. This Journal of Bioinformatics is the one specialized in this type of things. Although I introduced you to bioinformatics, the field of study that is concerned with dealing with chemicals using computer programs is chemoinformatics. Okay. We took all solutions through the research we did because we need to offer some service. And this is one of the causes that we have in bioinformatics. In bioinformatics, we don't care only about just a novel algorithm. Because if you go to the field of machine learning or some other computer science theory fields, they're focusing more on the solution. Here, we need a good solution, but at the same time, we need to find practicality with that solution. So we took all of the developed methods and we grouped them into a single system that you can access online. And that system, it's not so clear here in the slides in front of you, but it will have some instructions. You can just select the number of the data set and directly it will generate features, it will develop models based on the given options, it will do all the pre-processing needed. And not only this, so it integrates all solutions that we have, Graph, DWF, Drama. Not only this, it will take you to another page where you can give it actually a list of drugs. So you can now use the system to develop a model for a certain experiment and then use that model to know which drugs in the market may interact with that target. And then you generate a list of results. This would be a snapshot of the output in table shape where you have the target name, the data set name, and then the drug name, and then we give you an activity score. What's the probability that this will interact or not? And we generate some visualization graphs where, for example, we revisit again this medicine zone, and we find that it interacts with 2 data sets with certain scores. These are some visualization tools that we have. This system gives you accessibility to process actually 1.3 million data sets. So PubChem Bioassay database have 1.3 million data sets. You can just input any accession number of those, will take care of the rest. But these are just listed as chemical compounds with activity rate. And then it allows you to know activities for 90 million compounds available in the databases, or about 8,000 drugs that are available out there. This is the link, you can actually visit it and try it. And I will share the slides, Dr. Ahmed, and pass it on. So okay, nice. We addressed some problem for bioinformatics from the perspective of data mining. What's going to happen in the future direction towards this drug discovery program? Remember that our ultimate goal is to reach this stage of personalized medicine. And many efforts nowadays is going towards that direction. And cancer treatment is on the table of really the top conferences in bioinformatics each year nowadays. So each year you go to the top conferences, you will find many works going on in cancer treatment. It is a big challenge that we want to undertake, and personalized medicine could be a key way to approach that. These give you 2 exercises. And the first exercise will give you the data variability. So if we are accessing all information that's related to chemicals and proteins, that's it. I have only chemicals and proteins. But then what about the size of the experimental results? Which means I have a small data of chemicals and proteins. Then possibly I can use that to do what's called drug repositioning. So So I know that this drug interacts with few targets, and then I have another drug that's very close to this drug. Maybe I can do some inference and say, okay, it can interact with one of the targets. But if we have so much data, we can maybe talk about what's called drug discovery. So the work that I did fits actually here. I was dealing with chemicals and proteins, but huge amounts of data. And that's helped a bit towards having some insights about drug discovery. Me and my friend Wael, who was a PhD student also with me, now he's a researcher in Toronto, we developed this method where we had few drugs and few targets. It's based on a graph mining, so we did not use the traditional machine learning techniques. We used graph mining to really also build some conclusions about the connections between drugs and targets. Now, in the near future, where I'm serving as a researcher in McGill University in Montreal, we'll be working on more types of data. Not only chemical and proteins, we will incorporate molecular information that we have from the human body and try to understand what's really going on and give more conclusions about the disease treatment. So of course this work was not done by myself. I had a very active group of colleagues working with me. I have to give a huge credit to my advisor, Professor Mayor Bejic, who is the director of our center there, who is really giving excellent support for us. I worked closely also with Professor Franz Kalnis, he's a known guy in the database field. I worked with my friend Wael, he was doing also PhD in computer science, and with Marquez Zaki, he's a biochemist, we had to deal with biochemists, and Dr. Mahbouba Ishaq is also doing biochemistry, and all the members. So again, thank you for giving me the chance to give you an idea about the research that I did and that I will be continuing to do. And here I finish the talk, and I welcome any questions from you. Yes, please. If we just have? Yes, we have just few points. Yes, so there are ideas, as I said, to enhance the representation of these few samples. So we can take some of the samples. I give a very easy solution. We have 10 points, just replicate them. Just copy them and duplicate the data set. It will be 20. Originally. But you can add some variations, which you end up doing, like you have the same point, but with a slight variation. And this way you are assuming this is the space of these points. So these are basic, simple ideas to just keep growing the... this is something you control. Yes. This is a good point. You can do some ranking of those features.
For example, you choose, for example, fuzzy erosion, for example, for modeling and the feature there.
Yeah, this is certainly one of the ways. So feature selection is a problem with many approaches to overcome. One of the strategies is indeed to rank the features. But I believe ranking the features would mean dealing with the features individually. What we were doing here, we wanted to know a group of features that they have good impact on the performance at the same time as a group of features. That doesn't mean that you cannot do ranking as a different way to do it. So ranking the features would probably mean that I need to test in a way that could be local. And here we're doing global weight session. Because when we're doing ranking, we will take one feature and see how it impacts the system, add the second one, the third one, and the list. But we compared with one of the solutions that we're using a similar strategy. Like MRMR, method, GMI, they were ranking the features, and then we were taking the top ranked features and comparing with our selected features.
And the other one for the overlearning for the algorithm.
Yes.
You choose a specific method for
We use many things. Actually, the paper shows very comprehensive experiments where we take the genetic algorithm and we try over all the data sets with different mutation rates, with different crossover rates, maybe with different stochastic conditions, with different number of generations. And we were keeping all of that as a way of
with the solution
well, certainly we have to choose some parameter based on the figure, right? But can we call this optimal enough? So it's tricky because we are dealing, if we are having some function to optimize and we are just running genetic algorithm, we are fine. Just wait it to converge somehow and choose your optimal solution. But here, the convergence is not straightforward. It might converge at a certain point, but in the actual practical case, it might miss actually many of the cases. So when we're dealing with these machine learning algorithms, we need to be a bit careful on how we're optimizing them. And the strategy that we're using, are we using all out, cross-validation, error, cross-validation, how we're caring about these things, because they will affect the model selection that we're doing for the specific algorithm. Yeah, so but we experimented with many generations and we found some optimal numbers for our own data sets. But we give actually in the paper some conclusions for the different data sets. Like if you have some kind of similar features, sometimes you might use such parameters. But in general, it's difficult.
Really would like to thank you for your interesting presentation. ما شاء الله احنا الحضور حتى كان ما شاء الله 10 مرتين ما شاء الله. I'm very interested in your presentation. No comments, that's it. لا no questions.
ما في أسئلة. يعطيكم العافية دكتورة وسعداء بحضوركم. جزاكم الله.
إن شاء الله. شكرا لك.
You did mention that data acquisition, so data mining. So where is the knowledge graph?
Where is the knowledge graph?
The second question is how you validate your model.
Okay, fine.
You compare the results with that.
Okay, good point. I think a little bit the two questions are related because we have data acquisition and then we do data mining. Now, where is the knowledge? The knowledge comes when we are saying that this drug would interact with this target or not. So we were giving these conclusions. We were giving this knowledge because this is what we care about. Are we really having some type of interaction or not with that specific cases? As for the experiments, yes, I've showed in a few experiments that we are comparing always with the best reported state-of-the-art methods so far. And not only this, as I showed you in the last example, that was not sufficient because we are working in bioinformatics to say that I outperformed some other methods. So we went further and we developed a case study where we studied carefully and closely one of the diseases and we checked that medicine. And I gave my conclusions and my knowledge that I got through the system to biochemists. So they're specialized scientists who came and they did independently their own confirmation, and we included this part actually in the paper. So the papers that we wrote, we say that this makes sense from the perspective of biochemistry. Does it make really sense to say that this drug interacts with this protein? So they go and they check further the existing literature and the existing types of interactions, and they were giving some confirmation for some cases, which was good indication.
I'm familiar with the health informatics, bioinformatics. Using how to apply IT technology to solve a certain problem.
Sure.
Right? What is here, because we apply knowledge management. So we put that data in database now. So
Yes.
how we compare this, how we can make sure the study is the value result, the good result.
So maybe we can talk about this because it depends on the case. So sometimes If it's a database, we are having some challenges to talk about the data cleaning and the data acquisition process itself to know the data that we have are sufficient or not. What I'm talking here in this part of mine is about really revealing predictions, unknown predictions. So it's not just a knowledge that's retrieved from existing transactions. It's a knowledge that we build for the future. But if we're dealing with these predictions and we want to know, are they accurate enough or not? So we have some ways to validate that, given the field that we are dealing with. So in some fields, maybe in health informatics, we can get some ways to validate some information.
So I mean, then you're proposing that solution. You're proposing the new model and the results. So,
Yes.
You work from computer science lead.
Yes.
How do you decide the cost?
Yeah, I give, I think, a couple of comments on this. They don't trust me. They don't trust us. Simply. What
So this we can solve this by
we try to solve it by
knowledge management part. By knowledge sharing part.
yes, sharing is the
Knowledge harvesting part.
these are ways that I would say that could serve the purpose. I agree. And I think additionally we need to bridge the gap between us and them, those scientists, those medical science, by talking more with them, discussing, showing them what we have and getting from them what we have. And one of the ways, as you mentioned, because this is one of the things that we did, right? We show one of the other methods where we check other existing databases. So let me take you to this figure
So
here.
we're dealing with the human heritage. We have to be very careful with the ethics, with this,
You are totally right. I just agree with what you're saying. But I'm not saying today you take this drug and use it for this
I'm
disease.
talking in general.
In general, this is a challenge that we face. Indeed. So what we did, if you want to take your terms and map it, so this was a prediction of my system. But then I went to one of the existing databases, curated databases that provide some other type of knowledge there. And we used it to see if there is, if we give it as a query, this drug and the disease, if we see some relationship. And this is some way of proving the things, as well as working with the biochemists. Certainly, one thing that we can do that I did not mention here is taking those predictions and actually go and put them in the real test and test them in the laboratory. But this could be in the foreseen future. But we have this is was not right now in our plan. But we could do this in the future. We can take some of those predictions, send them to one of the existing laboratories, pay a couple of $1,000 and get to know if they are actually working or not. And this is one of the ways to do it.
Can you go to the slide with the data acquisition? The second or the next slide.
So actually, I think in more one slide. So here I mentioned data collection. Okay, this one. So this, not this one.
Start from the first, please. From the first slide. Yes, there is something missing here.
What is missing?
Definition of bioinformatics.
Why are you saying so?
This is the definition of biology.
Yeah, it's a combination of computer science and biology
Yes.
and sector.
Yes.
I think the IT is missing.
We can't put IT. But this is one of the accepted definitions. It's not my own definition. And keep in mind, when we say computational science, they are not ignoring IT. It's just so when I say computational science or computer science or data science, I think management of information systems, IT, databases, management system, database research, different kinds of things fit in that. I think this is what it's meant. It does not mean to ignore that, but to shed the light on three directions. So we have computational sciences, we have biology, we have statistics, and three main fields that interact with each other.
It's the same definition of informatics, we just add biology.
Yes. Yes. Could be, yes.
Data is data acquisition. I think the next slide. Can we what do you think if we have a place with knowledge acquisition?
Which one?
One who started with this sometimes. Data acquisition?
Say data collection.
Data acquisition. How do you do that? Manually? Do you do that manually or use the AI computers?
No. We use computers.
Okay, we're running out of time actually.
I'm at your disposal.
30 minutes and 10 minutes for question. I would. Is there any comments, please?
I have one curiosity. In the past 2 or 3 slides, within past 2 or 3 slides, I saw that you in your center, the journal papers are more than 400 plus, 400, but the conference paper is only 25.
Yes.
Usually, yes. Usually the conference papers are more than the journal papers.
Professor Masoud? Masoud?
Yes, I apologize.
Professor Masoud. Indeed, this is right. And if you realize, through my PhD, I only publish journal papers. So this depends on the field. In bioinformatics, they focus, their venues are actually the journals, contrary to the computer science venues, like machine learning venues, database venues, where the best venues are actually conferences. But in bioinformatics, we talk about journals. There are few top conferences, but still the focus is on journals. And when we go through the hiring process in these places, even outside, they focus on which journals you are publishing in.
I think it should be the other way around.